GM1 gangliosidosis does not have a currently approved treatment that would alter the natural history of the disease. Most of these families receive the diagnosis, and once they receive the diagnosis, the only option available is the current standard of care, which is comfort care, minimizing the complications of the underlying disease, trying to manage the comorbidities that accompany GM1 gangliosidosis, and slowing down or reducing the discomfort that results from the inevitable. But eventually, these young children, they do succumb to their disease, especially the early infantile children at an age between 2 to 3 years, and there is no treatment available at the moment. PBGM01 is an AAV-based gene therapy. AAV is short for adeno-associated virus. It is a virus that does not cause or is not known to cause any disease in humans and has an excellent safety profile. It's been used in numerous clinical trials to date. The AAV U68 is a capsid that we use for our Imagine-1 clinical trial. It is a variant of the serotype AV9, which is known to have an excellent distribution in the central nervous system. And it carries a full-length human transgene to deliver to these children who have a defective gene resulting in the lower enzyme levels. This year at World 2023, we did share updates on our Imagine-1 clinical trial for PBGM01. Now, this is a phase 1/2 safety, tolerability, and efficacy study assessing the PBGM01 administration through the intracisterna magna in a specific patient phenotype of GM1 gangliosidosis. And our patients are either in the early infantile or late infantile patients population. In December of last year, we released data on cohorts 1, patients 1 through 6. And one of the things that we obviously focus on in a phase 1/2 study is how well tolerated was PBGM01. And we're very encouraged with the safety profile. It has been very well tolerated generally across all of the patients in the sense that we have no treatment-related serious adverse events. We have no evidence of what has been previously described in preclinical studies as AAV peripheral neuropathy or dorsal root ganglion toxicity. We have not seen that based on clinical studies or our nerve conduction studies. And in addition, we have not had any complications related to the ICM administration itself. And finally, we have a favorable immune profile with no antibodies to the transgene product. In terms of the efficacy, one of the things that we're very excited about is building on the dose-dependent response that we reported on. So in our key biomarker data, we reported on the dose-dependent response with the higher dose that we saw in the expression of the transgene. So we saw elevated CSF beta-galactosidase levels in our children who received the high dose in a dose-dependent manner compared to our first dose. Now, with that, correlating with that, we saw a reduction in a dose-dependent manner in one of the GM1 gangliosides that are measured in the CSF. So that was very encouraging. And as new data this year, we are presenting additional data clinically and in our biomarkers. This year at World, we are sharing new data from a clinical standpoint, and that's the MRI severity score on our patients. The MRI severity score is a score that was developed and validated mostly in late infantile and juvenile GM1 gangliosidosis patients. It is a score that is based on cerebellar atrophy, but also white matter abnormalities. The higher the score, the more structural damage there is in the central nervous system. And one of the things that we find to be very encouraging is that across all of our cohorts, we do observe stabilization of the score in these patients, as opposed to what's been described in the natural history for late infantile patients, that the score rises over time from the time of onset of symptoms. Now, the duration of follow-up, we will continue to see and continue to assess this score for our patients throughout the study. Also new at Worlds this year, Additionally, we have shared new substrate data. We are sharing our urine DP5, which is one of the substrates that we are measuring. The observation was that we are seeing prominent reduction in the substrate levels, supporting the observation that we have reported on of peripheral beta-galactosidase activity.